I changed I kept the title that you will find in your folders, but I also changed it slightly into a less technical explanation, which better describes the concept that we are following in the last years. And that would be fighting prions with the prion protein, or better to say, with prion-derived fragments. But where do we have to? Coming from Germany, let me shortly thank the German national soccer team, because as they decided, for whatever reason, to not go that far in the current World Cup, I had not a single conflict of interest in coming here instead of doing a public viewing of the final tomorrow. But of course, there's more important things than soccer, and here are the people that I really would like to thank, and you see it on the bottom of these slides, and these are, of course, the donors for the grants that I received. Not only the donors of this year's grant on the right, but also the donors, the families of the grant I received 2 years ago. And this slide also shows you, in principle, our concept that was the basis for both projects. So in the middle you see a schematic representation of the cellular prion protein, so the good guy that is anchored to the cell membrane of our neurons and other cell types as well. And since several years it is known, just a remark, so for the rest of my talk, the green guys will be the good guys, and red guys that you will see later are the bad and evil guys. So since several years, it's known that there is cleavage events that occur constantly in all of our brains right now. And on the right side, so cleavage events by proteases, and I depicted these proteases like small Pac-Mans here. You can also imagine them as molecular scissors, because this is exactly what they do. They cleave proteins at specific sites. And on the right side, you see the so-called alpha cleavage. I will go on in more detail later. And this cuts in the middle of the prion protein, and it releases the unstructured, please keep that in mind, so it's a flexible, non-structured domain. The N-terminus from the prion protein releases it into the extracellular space, while the folded C-terminal part, the other half, stays attached at the plasma membrane. And on the left side, and this was the research grant 2 years ago, on which we are continuing to work, this is a so-called shedding. As you can see, it takes place much closer to the plasma membrane and more or less releases the full-length prion protein into the extracellular space. Now, it turns out, and we and many others believe, that these cleavages may act protective in neurodegenerative diseases. What you see here in red, one is the so-called A-beta peptide, or in an oligomeric form. This is the bad guy in Alzheimer's disease, or one of the bad guys. And the other red group there is, as you know, the misfolded prion protein. So it's known that these have to interact with the cellular prion protein at our membrane in order to transmit toxicity, and in the case of prion diseases, to template the misfolding. Sorry, and just to remind you, because or to give some flavor of our concept might hold true, this is now for the shedding. What you can see here on the bottom is a Western blot analysis. What we are doing there is comparing protein levels, and here we did it in 3 different animal models, so in the brains of animals. And basically what you can see here in the upper part of this Western blot is that if you have mice that don't have the protease ADAM10, the orange guy, the orange Pac-Man that you can see here, you don't have any shedding of the prion protein. In the middle you see normal mice, wild-type mice. This is what is constantly produced. And on the right you see a mouse model that has overexpression, so manifold overexpression of the prion protein, and you see a lot of shed PRP in these mice. And if you compare now this to the lower blot, which is showing you, because these mice were prion infected, and it's showing you the bad guy, so the misfolded PRP-SC, you can at least, I hope, appreciate that there's a strong correlation, or inverse correlation, I have to say, indicating that indeed, the more you shed, the less PRP-SC is produced. But I don't have time to go into detail into this, but we can probably discuss it later. So now coming to the actual project. And in contrast to the shedding, where we know what exactly is the pacman, what is the protease, here we don't really know the identity of the protease. And if you ask me, I even believe that there are several proteases doing it. So since we don't know what is the protease, we don't have any tools of pharmacologically targeting this one. Although it has been known that this alpha cleavage, in general, somehow acts protective in different kind of disease conditions. And moreover, and especially with regard to Alzheimer's disease models, it has been shown that these, the released N1 fragment is able, as you can see here on the left side, to block these toxic amyloid beta oligomers, to really bind them and keep them away from the cell and to transmit their toxic effects. Surprisingly, many studies have been done on the Alzheimer aspect, but no study, at least not in vivo, has been done on the role in prion diseases. And that's really surprising because, as I told you, the initial interaction of PRPSC and PRPC is thought to require the N-terminus. And this initial interaction then only allows toxic signaling, but also the later misfolded process showing here. So could it be that this N1 fragment, as shown for A-beta, is also able to block PRPSC? And we decided, for various reasons, to start with a proof of concept study generating mice. And these mice, on top of the normal prion protein, now they express additional levels of only this N1 fragment. And I hope you can see it here. So on in the middle you see a bar graph. As you might know, you first have DNA that encodes for your hereditary information. This has to be transcribed into mRNA, messenger RNA, and this then will be translated into amino acid chain, which actually will be the protein. And this bar graph shows you that we succeeded. So we really had mice that have higher levels in the messenger RNA. And on the right, again, you see a Western blot. Of course, we didn't touch the prion protein expression, which you can see in the upper part. But I hope you can all see that 4 versus 4 mice in this case, we have really succeeded in generating higher levels of this N1 fragment. So far, so good, but now comes the but. And one of the aspects I would like to keep in mind is, if you have a really close look to this blot, the lower part, you might see that there's a double band and not a single band, and this is one part of a later explanation. Because, of course, we didn't wait for long. So once we had enough mice, we immediately inoculated them with prions, because that would be the most obvious thing to check if there's some kind of protection. So what we expected is this gray curve that you can see, at least a prolongation in survival. What we and everybody here was hoping for would be a cure that all mice stay alive, which you can see on the upper part. But what actually was true and reality is, and these are the blue and the red lines for the wild-type control mice and for our transgenic mice, is they basically didn't show any difference in survival. So have we been wrong with our strategy? And the answer is partially yes. Because at that time, then we started to isolate primary neurons from these brains of these mice. And now we were analyzing the fluid, so the media surrounding these neurons, because that would allow us to really check for what we were interested in, namely released N1, right? And here, if you just consider the right part of this blot, now for the first time you see that in media we don't see a difference in the release of N1 if you compare wild-type mice with the transgenic mice. And of course, so it seems that we don't have secretion. We have higher production, but no secretion. And of course, we need N1 to be outside of the cells. At the very same time that we got these findings, it was end of last year, so December 2017, a German colleague, Jörg Tatzelt, published a paper, funnily enough, using the prion protein as an example, but having a wider implication in general protein biosynthesis. And I don't want to go into the details, but for a protein to be either positioned at the cell membrane or to be secreted outside of the cell, it needs to be translocated. It needs to be translocated into the ER, the so-called endoplasmatic reticulum, and to then be able to go the way through the cell and to the outside. And what Jörg Tatzelt and his group showed that normally, in order to be successful, a protein at one point in its sequence has to start presenting a structured domain, which is here depicted like a helical structure. If this is not the case, and please remember what I told you about N1 only, it's an unstructured or so-called intrinsically disordered peptide. If this is not the case, translocation into the ER and all what would follow is not efficiently possible. And this is exactly what we can confirm now. So my boss, since we were pretty disappointed, came and say, hey, see it from a positive side. You now have the very first in vivo model of this study. But of course, this is not at all what we aimed for. So when you're fighting, of course, when you're fighting, as you all know, you have to live with knockdowns. More important is to stand up again. Knockdowns happen very often in research, I would say. So can we overcome this problem? And knowing this, what we learned here, we immediately started to generate another mouse model. So we learned that we cannot express N1 alone. We need a structural element, and this is shown here on the left. So we decided to exchange the C-terminal part of the prion protein for the FC part that is part of all of our antibodies in our body. So we hoped that this would lead to increase secretion, and this is on the right side, it's only shown in cells because the mice are in the making right now, but it holds true. So we see increase, and the funny thing that we also see is we still see an alpha cleavage-like event. So it seems that whatever comes behind N1, the cells or our body has mechanisms to still cleave it, and actually this is what we wanted, right? We wanted more N1 in the extracellular space. But as I said, these mice are right now in the making, and I hope in a couple of weeks we will do exactly the same thing that we did. Once we have enough, we will inoculate with prions, and I'm optimistic that in this case we will see a difference. And then, of course, hopefully this will lead to further directions. So, this is already my outlook. As I said, we will do a detailed characterization of these mouse. As I said also, the prion inoculation we will start right away. And we will also continue to identify the identity of the protease. And regarding the other projects that I just mentioned, we have just in the last couple of months, we have received some very promising results. We had to learn, for instance, that if you want to stimulate shedding, it might be better not to target the protease ADAM10, but the prion protein itself. But this is something we can maybe discuss in more detail on the roundtable discussions. I think I can skip this summary for the sake of time, because everybody can go through it for him or herself. And again, I would like to really thank the donors of the research grants I received, but to all of you donating to any research grant that is given out by this foundation. And of course, I also would like to thank my co-workers in Hamburg and all my national and international collaborators, many of them actually having received research grants also in the past. Thank you very much.